Hi, welcome to your first set of guided notes of the year. As I am going through this presentation, you will be filling out your guided notes. It will also stop you in different places to make sure that you understand the content as it's being taught to you. So make sure that you're answering those questions the best that you can. Okay, so let's get started. So we're going to start off with actually what is science, and if you take a look at this little thing here, you can actually see the entire scientific method out. So that's pretty much what the entire lecture will be about. So what is science? The definition of science is the use of evidence to construct testable explanations. So you're kind of seeing what the difference is here between science and pseudoscience. Pseudo means fake. So science is when you're actually using the scientific method to come up with your data. Pseudoscience would be you're not collecting data in the scientific method way. Science only deals with the natural world. It excludes supernatural phenomena. Unfortunately for me, I'm really big into like essential oils. That would technically be in the pseudoscience because there has not been any studies that showed reliable data showing it that it shows any results. I still like it. Important to collect and organize data in an orderly way, looking for patterns and connections among events. This is something that in our society we should do a lot more of, is making sure that we go through and we do things in organized and collect things before we come up with a conclusion. So science, make sure you fill out all the guided notes. Okay, so scientific methodology, or the scientific method, a general style used to investigate in science. So you ask your question, you do research, you create a hypothesis, you test with an experiment. Is the procedure working? Yes or no? If it's not working, maybe go back. Okay, if it is working, analyze your data, draw your conclusions. Were you right or were you not correct? So, observation, what's your question? Active noticing and describing events. This will lead to questions that will lead to your experiment. Do you have a question? Like today for the bell ringer, we asked, does fertilizer help a tomato grow? What if you didn't use it? That was my question. So hey, let's do an experiment to figure it out. Your question or observation will lead to an inference or a hypothesis. Your inference is a logical interpretation on what scientists already know. So you're inferring. Some people think that inferring, if you infer something, it's just like, it's got to be logical. Your hypothesis is a scientific explanation that can be tested by further observation or experimentation. So now you're ready to make an actual explanation. You're making your guess, hypothesis. When you create a hypothesis, you always want to make it if, then. If fertilizer is added to tomatoes, then it will give you a higher yield, or you will get more plants, whatever your dependent variable would be. Okay, so let's talk about our variables. So very important, sometimes students get these confused. Your controlled variable means everything stays the same. So within your two groups, which we'll get to in a minute, it's something that you're not changing. So it's not changed. So all variables that are unchanged and controlled by scientists. Only one variable should be changed. If you're changing more than one variable, you won't know what's working. So if you're adding extra water and more fertilizer, how would you know which one was working? So you would want to keep the water the same amount of sun, everything controlled. You are the experimenter, you control everything. You have your independent variable. That is what you are manipulating or what you're deliberately changing. That would be the amount of fertilizer. So this one has fertilizer, this one does not. That's what I am testing to see. Your dependent variable is what is the data that you're collecting. So it's your response to the independent variable. Did it work or not? How tall your plant is. The amount of plants you get out of it. The amount of tomatoes you get out of it. It's depending on how you want to determine that. But that is a difference between independent is what you are deliberately changing. Dependent variable is the response. Okay. Control and experimental group. So your control group is not exposed to anything. So you always want to have a control group because how would you know if it worked or not? So you'd have one control group, no fertilizer, right? So one control group would have no fertilizer in it at all. You would just add the same amount of water, get everything else will be controlled, right? So how much sun, everything will be the same. And then you would have your experimental group. Your experimental group would be exposed to all conditions, including now independent variable. So the fertilizer. Your experimental group, you would have fertilizer, control group, you wouldn't. Okay, does that make sense? So those are your different groups. The data. So now you've done everything, right? You've waited, usually takes about a month or two before you really start to see some good yields in the tomato. You'll have either quantitative data or qualitative data. I'll give you an example for both for our big experiment for the fertilizer. So your quantitative data, numbers that you obtain by either counting or measuring, you need numbers, right? So the amount of tomatoes, how tall your plant is, you are actually writing down a number, quantitative. Qualitative data would be the quality. It's more of using your senses. So you would be like, oh, it's pretty, it's like a nice, pretty tomato versus these tomatoes are not round, they're a little brown, they're just not the quality that I was looking for. So that would be an example. We could say like, oh, this plant's kind of slanted over versus these plants are nice and upright. It just looks healthier. So quantitative would be like the number of plants versus qualitative would be you look at it and you observe different characteristics. Or the taste. That's a good one. The taste of the tomato versus like, is it good or not? I mean, that's probably the best thing. Is it healthy or not? Okay, so once you have that data, you got to draw a conclusion. What was your results? So you went back, you looked at it, you said, wow, I had 15 plants versus 3. This tomato, so your quantitative, these tomatoes taste really good, and these, I didn't like them. So that would be the difference. So data analysis may lead to a conclusion that support or refute your hypothesis. You'll go back to your hypothesis. Was I right or was I wrong? And that's where you would decide there. Okay. So we're going to switch it up a little bit. So theories. So how do people come up with theories? A lot of people think that a theory is like a hunch or I have a theory that it's almost like they're saying it's a hypothesis in a way. Like I have a theory that the tomato plants would be better with fertilizer. That is incorrect. A theory is a highly tested, so that means this has been tested using the scientific method over and over again. And every time it supports the hypothesis. So there should be more emphasis on theories. Yes, we say sometimes it was just a theory so that, but no, like if it's a theory, that means a lot of quantitative and qualitative data has come back to support your data. Darwin had a hypothesis about evolution. He went and saw some finches and noticed that each finch had different beaks depending on what they were eating. So he said, hey, I think all these came from the same ancestor, but then due to adaptation, they changed the size of their beak and the shape of their beak. So that was, and then he did a lot of different data, quantitative, qualitative. Okay, switching again real quick. Biotechnology is something that kind of leads off the scientific method. We kind of talked a little bit about this, but the application of technical process, invention, or method to living organisms. It could be agriculture, so how we make food. Genetically, modification leads to better, less expensive, and more nutritious foods. I think it's funny how some corporations have jumped on the back of, there's no GMOs. I mean, just because you're moving around some DNA doesn't mean it's bad. Sometimes it does, though. Health and medicine, source of medical advancement and prevention of disease, and cloning goes in there. Some more specific examples, if I went a little fast on that, just go back and pause. You're allowed to rewind. Genetically modified is usually abbreviated with GM. It leads to better, less expensive, more nutritious food. I already said that. An example, they will actually put DNA to resist insects. So corn doesn't even have to be sprayed with insecticides, which is actually really bad for you. Insecticides are, but the DNA shouldn't be too bad. So you don't have to spray it with chemicals because it's already embedded in the DNA to prevent it. A really weird example my professor told me in college was they will actually put in tomatoes a strand of DNA from a spider that is resistant to cold temperatures. And that is why some of them don't get, they don't die in cold weather. So sometimes you might be eating some spider DNA. Animals can be genetically modified. This one's a little sad. 30% of milk comes from cows that have been genetically modified to increase their milk. Preventing disease, medical research, and forensics. And I do believe that's the last slide. We are done. So hopefully you got a lot out of that. You filled in all your blanks. If you don't like your scores, you let me know. I can reset it. Otherwise, great job. Make sure you get 100% on it.